Hello, my name is Gunnar Rätsch. I'm going to talk about the SPHN-PHRT driver project, Swiss Molecular Oncology Breakthrough Platform. So this is a project together with Mark Rubin, who is the PI of that project, who cannot be here today, and I'm presenting this project. So let me give you a one-slide summary of that project. So this project has two parts. One is the SPHN driver project, and the other one is the PHRT research project. So the SPHN driver project is led by Mark Rubin, and the PHRT project is led by me. So the main question will be for the SPHN driver project to create a breakthrough clinical genomics platform to manage, share data across Switzerland to help the clinicians and scientists to untangle the complex therapy-related resistance. So we do this together with the international research community. The PHRT platform will use the SPHN driver project to test and address an important cancer immunology question. In particular, we would like to understand why do certain patients respond to drugs blocking immune checkpoints such as PD-1 and PD-L1 pathway, and what is the potential mechanism of response and resistance. So these are the key questions, and both parts of this project work together to address this. Let me go a bit into the organization of that project. So the project is led by Mark Rubin and me, and we closely interact with Holger Moch from University Hospital in Zurich and with George Kukos at EPFL. So we work very closely with the molecular pathologists at the 5 university hospitals. So we have one expert pathologist from each of these hospitals. We also work with a core bioinformatics team from Bern and also one from New York. And we have a strong coordination team to bring the organization together. So, as Olivier mentioned, and I'm grateful for this introduction he gave before, we closely interact with the Partner Oncology Driver Project, led by Olivier and Mohamed. So there's many partners involved, as I said, all the hospitals, EPFL, ETH, and many research foundations which are funding this. There's many people involved in this project, and this is just the group heads of all the labs which are involved. So behind each of those, there is a research group or a group of people who work towards this project. So it's really a large-scale research project with a single aim or with a few aims which we have in common. So does this actually work? Can such big projects actually work? And we do actually have a lot of experience working in these very large-scale research projects. And one of these is, which I'm involved in, and also Mark Rubin is involved in, is the International Cancer Genome Consortium. So this is a very large-scale research project which has overall about 800 scientists. A sub-project of that is 200 scientists, and I'm leading one sub-project of this sub-project, which is 50 scientists. And we all try to solve very similar aims, and we use jointly some resources which we develop together. So, and in 2018, about 50 papers, probably in Nature, will appear, and this is a joint effort, and this is very successful. So, there's obviously some complications when you work with many people, but I think there's a joint spirit, and it's really amazing what you can accomplish if you don't care to get the credit, who gets the credit. And we all think that we are part of a large, great project. So, and this is really one of the principles and goals of this project. So do something great, support junior faculty and students, and change clinical and research practices. And obviously, we need to have fun together in order to be efficient. Okay, so this project's collaboration scheme really tries to bring together the hospital side with the ETH and university side. So the research, maybe academic side, with the hospital pathology side. So the SPHN part focuses on connecting pathology with bioinformatics to really make the bioinformatics as efficient as possible and as useful for pathology as possible. The PHRT part connects immuno-oncology with data science. Data science, we use machine learning, efficient data processing techniques, and we try to address the question in immuno-oncology. So here are the main aims of this project. So the first aim of the SPHN part is to find out the optimal configuration for determining a uniform genomics reporting and to enable sharing of clinical and research data. And this is done together with partners at SIB. So the second part is to harmonize the clinical genomics reporting across different hospitals. Right now there is a pathology report which contains clinical genomics information, and in every hospital this is organized in a different way, and it's very hard to it's not harmonized. And here we work together with the SNF Patholink project and also with the SPHN Oncology project. So in third, we need to we think about and develop new techniques for data sharing for research in clinical data. So we would like to establish multiple sites, starting with the 5 research hospitals, and to improve and use features for broader clinical and researcher connectivity. So the goal is that we have an integrated clinical report with very detailed interpretation and very detailed information. This will lead to better treatment of the patient, as we have heard before, harmonization across the hospitals, which will make it easier to exchange data. And we can also use this to collect observational data of thousands of patients to enable biomedical research. And Olivier has given a few examples on this. In the end, we have to do this using or we would like to do this using the Swiss Personalized Health Network and Data Coordination Center. The data from the hospitals go into a secure research environment, IT environment. This is where we perform the research, and then we can give this data back to the hospitals and to the patients. In the end, we can visualize and analyze such a Swiss cancer cohort and make perform research on such a system. So for instance here, the CBio portal, that is also what Olivier has shown. So the research part concerns the question, why do some of the patients respond to these immunotherapy drugs and why some do not? And what's the mechanism of the response? And here we have 3 work packages. One is related to sample collection and processing. The other one is an integrative genomics analysis. And the last work package is developing machine learning techniques and also validating a specific hypothesis which we have. So we are going to develop techniques to collect the clinical data. And here we work with SPHN, with the SPHN part. We will sequence RNA and whole genome via the Swiss Genome Center, which is co-funded by PHRT. We will perform mass spec on these tumors, also via the Swiss Proteome Center, funded by PHRT. And we will perform deep immunoprofiling and T-cell receptor sequencing via the Ludwig Center in Lausanne. One key idea of this project is, or one key question which we had in this project and which generated this project was a question connecting alternative splicing and cancer. And this is the result here of a study which we're about to publish very soon. And we looked into cancer of about 8,500 tumors, and we looked into splicing differences in tumor versus normals. What we found is that there's many differences of splicing of tumor versus normals, and there's much more alternative splicing which is happening in tumors than in normal samples. And it turns out that you find some samples where this is very extreme. You find here on the bottom, you see the number of splicing events per sample. 1 dot is 1 sample. And you find that some samples have really a large amount of alternative splicing which is going on. So the idea is that we can exploit this in immunotherapy, because these splicing events, they generate peptides, and these peptides can be targeted by immunotherapy. So the question now, which we would like to address with that project, is to can we exploit aberrant splicing in tumors or in cancer? Can we exploit that for immunotherapy, for targeted immunotherapy? And this motivated some of these designs of the project. So we have a validation phase where we try to address exactly that question. And we have a second phase where we go into 2 clinical trials with each 100 patients, where we take the tumor out of the we take a sample out of the tumor, let it grow ex vivo, treat it in vivo and ex vivo, and analyze RNA and whole genome sequencing for these tumors. And the goal in the end is to develop machine learning and use molecular insights to accurately predict immunotherapy success. So in summary, the Swiss Molecular Pathology Breakthrough Platform has 2 pieces: the SPHN Driver Project, where we create this genomics platform to manage and share data across Switzerland. And here we optimize and harmonize the molecular pathology reports. And we develop, using the SPHN infrastructure, we develop data sharing and analysis techniques. And this will enable novel research and brings breakthroughs. One of these projects which uses this platform is this PHAT project, which I talked about. And we will analyze immunotherapy mechanisms in patients. So we would like to understand the effect of alternative splicing in cancer and immunotherapy. We try to identify and combine molecular markers. And we would like to predict immunotherapy success with machine learning and translate in the end these findings back into clinical practice. With that, I would like to thank you for your attention and thank all the participants of this project for working with us. Thank you.